students, today we will study bacteria, the endospore formation. Endospores are highly resistant body produced within the cell of gram-positive bacteria, such as in the genera like Clostridium and Bacillus. Both are rod-shaped. The genera Bacillus and Clostridium have species that are important human pathogen. Their pathogenicity is considerably enhanced because they are capable of forming endospore. Endospore formation is not a means of reproduction since usually there is no increase in the number of cells during the cell cycle. A single bacterium formed a single endospore by the process known as sporulation. Endospore serves rather to tide the bacteria over a period which is not favorable to the vegetative growth. Endospore. The process of endospore formation takes several hours and is known as sporulation or sporogenesis. Endospores are inhibited when a nutritional deficiency occurs or the environment becomes otherwise adverse. The process starts with the replication of DNA in the cell to form two complete set of DNA. Within a short time, a small portion of the cytoplasm contains one set of newly replicated chromosomes. Some ribosomes are enclosed by an ingrowth of plasma membrane called spore septum. The spore septum becomes a double layer membrane that surrounds the DNA and other cytoplasmic material. This structure entirely enclosed within the original cell is called the forespore. Thick layer of peptidoglycans are laid down between the two membranes. This structure forespore undergo dehydration and contraction and then surround itself by many layer wall consisting of cortex, spore coat and exosporium. During the maturation period, the refractability increases in the endospore. At maturity, the endospore is liberated as a result of autolysis of the parent cell. Physical and physiological properties of endospore. Endospores are thick-walled, long-lived, and remarkably resistant to heat, radiation, chemicals, and other agents that are typically lethal to the organism. They may remain dormant for many years. This marks the resistance of endospore to heat, aging, and toxic chemical has been attributed to the chemical composition of relatively thick, impermeable spore wall and to mark dehydration. All endospore contain large amount of dipiconolic acid, a unique compound that is not found in the vegetative cell. It occurs in combination with large amount of calcium and is probably located in the core, that is the central part of the spore. The calcium dipicolonic acid complex may possibly play a role in the heat resistance of the endospore. Germination of endospore. If adequate nutrients are available, the endospore sprout or germinate and once again become a vegetative cell. Nutrition in bacteria. Bacteria can be divided into two main types based on the source of carbon: autotrophs, which synthesize food from simple inorganic compounds such as carbon dioxide and mineral, and heterotrophs, which obtain their food basic from carbon compound from the pre-synthesized organic molecule. Each of these types may be further subdivided on the basis of the type of energy source needed to reduce the carbon. The energy needed to reduce the carbon may be derived from the sunlight or from the chemical compound. Those bacteria which utilize light energy are called phototrophs, while those using the chemical energy produced by the oxidation reduction reaction of inorganic or organic compound are called chemotrophs. Autotrophs are also referred to as lithotrophs. Rock eating and heterotrophs are referred to as organotrophs. Bacteria can be divided into the following four nutritional types according to the manner of processing, procuring energy, and carbon source. Photoautotroph or photolithotrophs. They use the light as a source of energy and the carbon dioxide as the principal carbon source of carbon. Certain pigment bacteria are able to utilize the light energy in the fixation of carbon dioxide to carbohydrate. That is, they carry out the photosynthesis. The process photoautotrophic bacteria possess bacteriochlorophyll pigment that enable them to absorb the utilized light to in the manufacture of food. Bacterial chlorophyll and the chlorophyll A have different absorption spectra. The photosynthetic bacteria uses longer wavelength of light than do the plant. Furthermore, photosynthetic bacteria use substances that other than water as a reducing agent. Therefore, in bacterial photosynthesis, unlike plant photosynthesis, oxygen is not released. Anoxygenic, they cannot carry on photosynthesis when oxygen is present. Most photosynthetic bacteria live in an anaerobic habitat such as lakes or pond bottoms. Some of the common examples are green sulfur bacteria, the purple sulfur bacteria, and the purple non-sulfur bacteria. The green sulfur bacteria such as chlorobium and the purple sulfur bacteria such as chromatium uses the sulfur compounds such as hydrogen sulfide and the hydrogen gas to reduce the carbon dioxide and form organic compound. The reaction is the carbon dioxide plus hydrogen sulfide to form the CH2O, carbohydrate plus sulfur plus water, and the energy is released. The green sulfur bacteria are distinguished from the purple sulfur bacteria by their type of chlorophyll and the location of the stored sulfur. Bacteriochlorophyll of the green sulfur bacteria are found in the vesicle called the chlorosome, underlying and attached to the plasma membrane. In purple sulfur bacteria, the bacteriochlorophyll are located in the invagination of the plasma membrane. Thank you. Further, we will study about Volvox, the new genus